Hi guys, we'll start the class in just like 2-3 minutes in case anyone else wants to join. Yep. I hope all of you can see the screen. So we can start the class for today. So we'll pick up where we left off last time, which was we were talking about the digestive system, right? And so we had a few more questions that were left about the stomach and some important elements, cells of the stomach, and some digestive properties, and how these different cells and tissues of the stomach contribute to the stomach's function. So we'll go through that and then move to the newer topics that were introduced about the liver, the gallbladder, the endocrine system, then the thyroid gland. Let's see how much we can cover in today's class. And then, of course, if there's anything left, we'll continue on the next session. Okay, so let's get started. So as we know, last time when we were talking about the digestive system, two of the major um, well, two major regions in the alimentary canal is obviously where you start with the intake of food, which is at the level of the mouth, and one major gland that is very important for making sure that food is moistened and the taste is you are able to sense taste and digestion also begins at the level of the mouth. That is the salivary gland. So the saliva is a very important secretion in that aspect and determines many steps beyond. And then we talked about, of course, the stomach, which is at the end of the esophagus or the food pipe. And we talked about the several different types of very, very essential cells that are found in these sort of little the gastric sort of crypts kind of looking things. And these cells are essential for producing acid, for producing mucus, for producing um, even enzymes, right? So these are really important for that. So so this we learned about the salivary gland and we learned about how there are several different types of major salivary glands and there are these acini within the salivary glands where primary saliva is produced and that as it moves through this sort of duct, there are several reabsorption and sort of calibration events that make sure that the osmolarity of the saliva is just right for the mouth. And actually, the osmolarity is lower, so it's not the same as blood. So then, secondary saliva is what finally reaches our mouth after it has been sort of calibrated while it moves through this passage. And we learned about the different uh, components of saliva, amylases for digestion of starch and all, mucins that make it slimy. We have some antifungal, antibacterial stuff as well that protects us from any harmful stuff that might be there in our in the food we intake or anything else, and so on. So there's a lot of different important and of course immunoglobulins as well. Um, which are very important for protection at the innate level, and lysozymes, as mentioned, which are very important for bacterial lysis. So these kind of different components and buffering agents are all present in the saliva. And then we also talked about the stomach, so the basic anatomy. So these four different regions, the cardia, where the cardiac sphincter is there that connects the esophagus to the stomach. This fundus, this upper region, the body of the stomach, and the pylorus, where the pyloric sphincter is there that connects it to the duodenum. And yeah, and then we learned about the layers of the stomach as well. So these different layers, the mucosa innermost, then the submucosa, the muscularis externa, and the serosa, which is the outermost. And in the mucosa, you have these, as I said, crypt-like things. So these are called gastric pits, which then are followed by an isthmus, which leads into the gastric gland. So these gastric glands and sort of the the uh, the wall that sort of surrounds this lumen region that has these different specialized cells that we talked about last time, like mucous neck cells that are important for mucus secretion, stem cells that replace damaged cells, parietal or oxyntic cells that produce HCl and intrinsic factor, which is important for vitamin B absorption, chief cells important for pepsinogen and gastric lipase secretion, and your enteroendocrine cells that, for instance, release like the G cells that release gastrin and so on. So as we saw last time, parietal cells are very important for HCl formation and the secretion of intrinsic factor important for vitamin B12 absorption. And these control this, and they are also controlled via the hormone gastrin because they have gastrin receptors. Um, yeah, and yeah, so they also control the H plus K plus ATPase on the luminal side, and this also so there, that's how one proton is transported while uh, 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 an opposing of K plus ion is also transported, and chloride ions also follow the proton gradient via this channel. So that's how one H plus and and one Cl are entering into the lumen and allowing the formation of HCl. So this is regulated with the help of gastrin and other hormones. Chief cells, they initially secrete the zymogen form, which is like the inactive form, like pepsinogen, and then it is activated by the gastric acid, so the pH, and that makes it into pepsin, which is active. And then we learned about these really important cells, the neuroendocrine cells. So the ECL, or the enterochromaffin-like cells, they are the ones that secrete histamine when stimulated by gastrin. So we're hearing a lot of gastrin because that is a really important neuroendocrine hormone secreted by the G cells, which are a type of neuroendocrine cell. And this is very important for directly or indirectly increasing HCl production. So there were two ways that we discussed. The first was stimulating these ECL-like cells to release histamine, which then potentiates the parietal cells, which then produces HCl. And the other is by directly stimulating the parietal cells itself via gastrin receptors that are found on those cells. And both of these increase H plus K plus ATPase activity, causing more H plus to accumulate in the lumen. And then there were also D cells in the stomach that produce somatostatin. Oops. Yeah. So that is just a recap of the most important points I feel from last time's lecture. And then we can pick up where we left off with some questions. Okay. So, right. Which receptor on the parietal cells mediates stimulation by acetylcholine from the vagus nerve? So I haven't discussed this per se, but let me know what you guys think. So you can either say the answer in your mic or you can type it into the chat box and we can discuss. Okay. So let me know what you guys think of this one.
M3 receptor.
Yeah. Does anyone else want to give it a try? Does anyone feel like it should be something else? Okay, I have one answer here. So I can explain this one. So the actual answer is indeed M3 receptor, which is the B option. Okay, so why is that? Because acetylcholine, which is released from the vagus nerve, it binds to these M3 or muscarinic receptors. Remember, we had discussed them earlier in a different context. And these muscarinic receptors are on parietal cells. And these M3 receptors are actually GPCRs, right? G protein coupled receptors. And they once this acetylcholine binds, that leads to a further activation of a lot of intracellular signaling pathways, leading to increased like activity of the proton pumps, and then that can cause more acid secretion. So this is very important when it comes to the parasympathetic stimulation of digestion. So the rest and digest response is um, majorly dependent upon this, because when we eat, there is a parasympathetic stimulation that leads to via the vagus nerve that can then trigger acetylcholine production, because acetylcholine, like as a neurohormone, then binds to the receptors while the stimulus is coming, and that's what leads to higher activity of the proton pump, which leads to higher H plus and Cl minus accumulation and more HCl, which is important for digestion, right? Because somehow our brain figures out that, okay, we have eaten something and then we have to digest it. So it's not like we have constantly acid being produced in our stomachs, right? There we have a sleep wake cycle, we have a a cycle for when we eat and what is the optimal time with respect to our schedule. So that is all part of our circadian rhythm, and this is just one of those things. It's just why if you don't eat on time, you get acidity and stuff, no? Yeah. Anyway, here's one more question. So histamine that stimulates the parietal cells is released from which type of cell? So we were just discussing this, if you guys remember. So I'm getting all interesting different answers, so I'm excited to discuss this one. Does anyone else want to try? Yeah. So the correct answer here is ECL-like cells, which is your um, C option. So G cells, which is A option, is the one that secretes gastrin. Okay, so not histamine. ECL cells are the ones that secrete histamine, and they are quite near the parietal cells, and that's important because histamine acts on parietal cells um, because it's sort of like a paracrine signaling molecule, and it can bind to H2 receptor. So in the previous question, this H2 receptor you saw, no? This H2 receptor is there on parietal cells, and that's what your histamine binds to, and that stimulates the activation of the proton pump, whatever, the H plus accumulation and acid secretion. Um, so if H2 receptors are bound, it leads to an increase of CAMP levels, cyclic AMP, right? And that can then stimulate acid secretion. So that's like the intracellular messenger for that. Secondary messenger is CAMP. So that's why when people have these sort of gastric acid problems, right? Like they say now that somebody has some gastric problem and or someone has acid reflux issues and so on. Like they just have this constant acid issue. Then they can have sometimes they are prescribed H2 receptor blockers that block these H2 receptors for histamine on the parietal cells that prevents. So when the ECL cells secrete histamine, the H2 receptors are already blocked, so it doesn't bind, so less acid is produced. So it's like at the molecular level, that's how you would treat a gastric acid disorder, of course, based on a doctor's advice. Okay, so I hope that's clear now to everyone. We can try one more question. So we learned about somatostatin, which is released from gastric B cells. So what kind of major effect does it have on acid secretion? Let me know what you guys think. Yeah, awesome. So you guys got it. It is C option, which is that it inhibits both the G cells and the parietal cells. So it's a very important regulatory mechanism, and this somatostatin is actually a very powerful inhibitor of gastric secretion, and it suppresses acid secretion by either inhibiting gastrin release from the G cells or it directly inhibits parietal cells. So it's like a negative feedback mechanism that prevents like too much acid production, particularly when your gastric pH is very low. So if it's very acidic, then there has to be some way to make sure that it's not so acidic that there's damage or major harm to the body. So we always, anytime you have anything, you always have a negative feedback mechanism, a way to regulate something, to make sure too much of it doesn't happen in every system that's available, right? In your immune cells, you have inhibitory neurohormones, right? And so on. Like in every system, you have the opposite. You have something to make it calm down or whatever, right? So cool, great. Glad we're all on the same page so far. So let's move to the next question. So which drug class directly inhibits this proton pump, the H plus K plus ATPase in parietal cells? So I think this one would be pretty easy for you. The key word here is directly. Yeah, this one is pretty straightforward. So these are the proton pump inhibitors or PPIs. So you guys may have heard of like omeprazole, right? So that, I mean, people take it all the time when they get like acidity problems and stuff like that. So omeprazole is a common one. Doctors prescribe it sometimes with other medications that could give you acidity or gastric reflux. So you take it before. Again, it should be prescribed ideally. So these targets the H plus K plus ATPase pump, um, which is on the parietal cells. Yeah, and sort of pump is like it's inhibiting it directly. It's like you're getting at the final step that's there before the hydrogen ions are secreted. So it's quite effective. And you're not sort of messing with any other hormonal circuit as much. So whatever the upstream stimuli are, if it's gastrin or histamine or acetylcholine or whatever, it doesn't matter because the pump itself is blocked in this case. Yeah. So, okay. I think I've repeated a question here, but uh, how does omeprazole reduce gastric acid secretion? I think this is pretty clear now, considering I just revealed it. Yes, exactly. Yeah, so omeprazole reduces gastric acid secretion mainly by irreversibly inhibiting this proton pump, right? So it covalently binds and then irreversibly inhibits. So covalent bonds, right, is very strong. They're not easy to sort of reverse. So that's why it's covalent and irreversible. And if it's permanently inactivated, then acid secretion will only continue after the cell has actually synthesized new pumps. So the existing um, pumps at the surface are all occupied irreversibly, meaning until those are endocytosed or cleaved off or whatever, and there is full turnover of that protein, and the cell produces new, folds it, everything, and sends it to the surface of the cell. Until then, you have lower level of acid production. So that's very, very effective, actually. Yeah. Okay. One more question. So, intrinsic factor, which is secreted by the parietal cells, is essential for the absorption of which of these vitamins or minerals? Yeah, fantastic. So, you guys have got it. It is option B, which is your vitamin B12. So vitamin B12 is called cobalamin, right? Cobalamin, cobalamin, right? And it's very important because how intrinsic factor works is that it binds to vitamin B12 inside the stomach, and it makes sure that it's protected from degradation. And so there's this complex that forms between the intrinsic factor and the B12. So, and this complex travels all the way into the small intestine and goes till the ileum, right? The last part of the small intestine, where then there are some receptors that specifically allow it to get absorbed into the bloodstream. So even if you eat enough quantity of vitamin B12, if you don't have the right amount of intrinsic factor, B12 cannot be effectively absorbed. It will not reach the right receptors in the ileum. Not enough of it will reach. It will not be adequate, regardless of how many supplements one would take or whatever. So this complex is very important for it to travel to the ileum. All right, I think we have just a few more questions and then we can talk about a new topic. So, if you have a deficiency of this intrinsic factor, which condition could it lead to? Do you guys know? Okay, awesome job. So, everyone's right. It is indeed C option, which is megaloblastic anemia. So, how does this work? So, essentially, it's the same thing where if you don't have intrinsic factor, vitamin B12 is not absorbed. Now, vitamin B12 is very, very, very important for DNA synthesis, particularly in cells that are very rapidly dividing, such as in the red blood cell precursors that are there in the bone marrow, for instance, right? And so then without that, what happens is if these cells are dysregulated in some way, that leads to these very abnormally large and immature looking red blood cells that are just they don't function effectively. And so that is if you don't have enough red blood cells, automatically you have some kind of anemia. So this one is called megaloblastic anemia because at the blast stage itself the cells are humongous. That's why. So very large RBCs but quite inefficient at their job, because the B12 at the level of sort of the hematopoiesis only is not is the level is too low, and so there's all these sort of abnormalities that occur. So great job. um, now it's clear, I hope. All right, one more question. So this pepsinogen, right, secreted by chief cells, and it's converted to the active form, which is pepsin, and it's primarily done by which of these? Yeah, so I think this one you guys would be knowing quite well. So the answer is C option, which is your gastric acid. Because pepsinogen is inactive and it needs to be converted to pepsin so that it's able to do digestion of proteins. Um, and hydrochloric acid is what converts this pepsinogen into pepsin. And interestingly, actually, once you have pepsin formed, it can actually start this like positive feedback process. So if you have some pepsin, it can actually activate more pepsinogen molecules. So you have some pepsin that's like an active form, and then that can act on pepsinogen and make it uh, active. And so you sort of have a loop where you start with some and then it sort of dominoes like that until, whatever, some saturating point, I suppose. So it's like a positive feedback process that can help to accelerate the digestion process when it comes to proteins that are being digested in the stomach. Yeah, cool. All right. So last question before we move on. So which hormone, which is secreted by the stomach, could actually signal hunger to the brain? Any idea? Yeah, fantastic. So you guys are getting it really all right, which I'm very happy about. So the correct answer, yes, it is ghrelin. So that is a peptide hormone. It's synthesized at the level of the stomach. So it's the ghrelin is actually called like the hunger hormone, right? So when the stomach is empty, the ghrelin levels will rise in the bloodstream, and that then signals to the hypothalamus, which is in the brain, and then that stimulates these hunger and food seeking behaviors. So then after you've eaten, then when the stomach distends, essentially the stomach stretches a bit, right? Because it's full of food now. And some nutrient signals are also there, and these signals then talk to the ghrelin secretion and reduce ghrelin secretion. So then you terminate the hunger signal. So yeah, that's why it's called the hunger hormone. So it's produced in the stomach mainly, and it tells you that you're hungry, or it tells your brain to tell you that you're hungry, right? And it acts via the hypothalamus. Yeah, that's, I think, all I really need to say about ghrelin. You may have also heard about leptin, right? In this kind of context, you would have heard about leptin and ghrelin. So ghrelin we talked about just now, right? So it's something that essentially is like a meal initiator, right? It makes you realize you're hungry and then go and get food, right? Because your stomach is empty, so you get signals. And then via hypothalamus, you're like, okay, I'm hungry. I got to go get something to eat. Now, leptin is a satiety hormone. So what does that mean, right? The main thing is that it suppresses appetite. So it tells your brain that we got to stop eating now. So it's actually leptin is not produced in the stomach, unlike ghrelin. Leptin is produced majorly in adipose tissue or fat tissue. So this also acts on the hypothalamus. So there are some certain neurons, and it acts on these neurons and promotes satiety. So typically after eating, or in people with like higher body fat, maybe, um, it the level of leptin is higher. So it's important for these kind of energy balances, and essentially it's like a sufficiency signal, right? You're getting from the body, where it's like, okay, you have eaten or you don't need to eat as much, and so this is what you should be doing. You should stop eating now. So these two work opposite to one another or one after one another. Um, to one would increase your appetite or make you hungry, the other would decrease your appetite or basically signal you to signal that you don't need to eat anymore. So it's very important for meal control, right? Because if you can't figure out, like, unless you physically feel sick from eating, how do you know that you're full, right? How are you telling that you're full? So um, these two hormones work together to control when you get hungry and when you get full. Yeah. Also, interestingly, if you think about it, in one clinically sort of relevant thing here would be like obesity. So, just a second. Yeah. So in the case of obesity, what happens is that amongst other many problems, there is high leptin because you have so much adipose tissue, so that the person, the obese person's adipose tissue is secreting tons of leptin. You have lots of leptin. So then why is it that that person, and this is obviously there are many different types of obesity. It's not always because someone's eating a lot. It could be different reasons, other medical reasons as well. But what happens is that actually that person's body starts become resistant to this high leptin. So the brain and the hypothalamus are not responding correctly to this high leptin, and so that satiety reflex is just not balanced properly. Yeah. So cool. Anyway, that's just some tidbit of information. Now we can move on to the next topic. So that's the liver. So we all know about the liver. Okay, so before this, does anyone have any questions or anything they want to discuss with me? You can let me know. All good? Then let's move on to this topic, right? So as you all know, liver, highly important, regenerative organ in our body, and liver is humongous, right? It has many lobes. Um, and it's a very, very, very essential, beautiful organ that does all of these different functions, right? So it makes bile, it helps carry this waste, and it breaks down fats into smaller bits in the intestine, which allows for digestion. So very important, which function of bile. It makes a lot of different proteins, important in the blood plasma. It makes cholesterols and special proteins that help carry fats throughout the body, because carrying fat is actually quite a task in the body. Um, there's a slight there's a storage of glucose, right, in the form of glycogen in the liver, and it can release it if so required. So it's like an emergency storage of glucose. And then, oh yeah, it also processes hemoglobin, very important, and uses the iron, because iron is important for many different functions. The liver actually stores iron. Also, the urea cycle also takes place in the liver. You guys would have studied it in biochemistry at some point, right? So this changes ammonia to urea, and that's one of the end products of protein metabolism, and it's excreted out of the body. Ammonia is much more harmful, but converting it to urea is quite important. It also clears the blood of medicines. So they say a lot, right? That doctors say, or like you hear this a lot, that okay, if someone's taking a lot of medicines, it does put some amount of pressure on their liver. Why? Because the liver is important for clearing out these medicines and foreign molecules that you're sort of introducing into your body for your medicine. So, or people who have long-term like drug abuse, they also can get liver failure. People who drink too much alcohol, they could also get liver cirrhosis, because it's all sort of detoxified by your liver, and if you put so much pressure on the liver, then the liver could really give out. Yeah, it also is important for blood clotting. It fights infections actually, and very interestingly, you might have seen people with certain diseases maybe who have like yellowish skin and eyes, and that's because there's like an imbalance of the amount of bilirubin that's accumulating in their body. So clearing the bilirubin is important, and if there's some problem in the liver, then that person could like look very sickly and yellow because of this accumulated colored bilirubin, right? So in the liver, when you say take a microscope and look at the tissue sections, you would see the cells of the liver, which are called hepatocytes, right? And these are arranged into lobules, right? Which are the structural units of the liver. So you have these lobules or hepatic lobules, and each lobule is sort of hexagonal in shape and drained by something called a central vein. And so if this is the lobule, and then you have so how does this work, right? How are these supplied with blood? It's a branch of the hepatic artery. Do I have that here? No, I don't. Okay, so there's a branch of the hepatic artery that enters into the liver and eventually branches into an arteriole, which then goes and uh, supplies this lobule. Then you have a venule, obviously a branch of the hepatic portal vein that's entering the liver. And remember about the portal vein thing. We'll talk about it in a minute, but yeah, and so then this arteriole and venule are both there at the lobule, and then you have the bile duct, which is very important. It's a branch of the bile duct that's leaving the liver with the ready bile. And the portal triad also has these lymphatic vessels, and it has fibers from the vagus nerve, which are parasympathetic, and it has this very unique blood supply, right? So you would have heard about the hepatic artery and the hepatic vein, but there's also another little system within, like a little smaller circuit of blood called the portal vein system. So the liver receives blood via the hepatic artery proper, but only like 25% of it, right? And it supplies all these non-parenchymal structures of the liver with arterial blood, and that is derived from the celiac trunk. However, there's another one called the hepatic portal vein, which is transporting about 75%, and this supplies the liver with partially deoxygenated blood that carry nutrients that were absorbed from the small intestine. And so this is actually the dominant blood supply to the liver parenchyma, and this allows the liver to perform its gut-related functions. And finally, the venous drainage of the liver then is achieved through these hepatic veins. And the central vein of the hepatic lobule forms the collecting veins, which then combine together, eventually form multiple hepatic veins, and then these all then go and open into the inferior vena cava, which then goes to the heart. Right. So that's about the liver. Yeah. Now we can talk about the gallbladder, another very important organ. Though people do survive without it, I know people, and you all know or have heard of people who have undergone like a gallbladder surgery where it had to be removed for whatever reason. You could have gallbladder stones, gallbladder cancer, anything. So, what is the gallbladder? It's an organ in the GIT, and it is over here. So, sort of below the liver, next to the stomach. It's pear shaped, and its main function is to concentrate and store bile. So I think it can go up to like 200 ml, and it's produced by the liver. So this bile is produced by the liver, and the gallbladder just sort of does some more quality control, right? So it concentrates it and it stores it. Um, and as part of the gustatory response, which is basically like you taste something, and that triggers this response of eating, like that you want to eat. So basically, a gustatory response is if you take you eat some food, then what happens after that, right? So those are different responses called termed as gustatory responses. And in this case, the stored bile that's there is released from the gallbladder in response to this enzyme cholecystokinin. So yeah, so as part of the hunger response or gustatory response, a bile is released from the gallbladder. And finally, this unpronounceable word, essentially it just means gallstones, right? I think cholelithiasis, maybe that's how we say it. Right, so that is basically, you guys would have all heard gallbladder stones or gallstones. These are basically just little lumps of bits and bobs, right? Like cholesterol and bile salts and whatever, and this stuff deposits in the gallbladder inside it. And they're pretty common and they can be asymptomatic, but they could also sometimes cause like a great deal of pain. And if they are like really sort of big, they could cause all sorts of, aside from pain, they could also cause dysfunctions. It could cause tissue damage. So then, in those cases, I think doctors typically remove the gallbladder altogether try to do some repair laparoscopically, but if there's like by removing the stones, but I don't know which one is more common. Yeah. And another concept here that's part of the anatomy of these structures is really important, which is the biliary tree. So it's just basically a bunch of ducts in the GIT that allow this bile that was synthesized newly in the liver to be concentrated and stored in the gallbladder before it is released into the duodenum or the small intestine. And so while bile is secreted from the liver and then it drains from the liver via these little canaliculi and these different kinds of ducts, left and right hepatic ducts, ultimately going into those, right? These ducts, they form the common hepatic duct, and that's very important because it runs alongside the hepatic vein, and as it descends, it's joined by the cystic duct. So you can see this. This is the common hepatic duct. This is the cystic duct, and it's joining over here. Sorry. And at this point, when the common hepatic duct and cystic duct combine, this is called the common bile duct, right? So common hepatic duct coming from the liver, cystic duct coming from the gallbladder. They join together, and everything after that, the melange of these two things, is the common bile duct. And then this descends further and passes evidently posteriorly to the first part of the duodenum, or the first part of the small intestine, and to the head of the pancreas. So when it reaches here, it is joined by the pancreatic duct, the main one. So this makes it the hepatopancreatic ampulla, right, or the ampulla of Vater. And this empties into the duodenum via some major duodenal papilla, and this papilla is regulated by a muscular valve called the sphincter of Oddi. Okay, so what are all these different terms we've learned so far? We have the common hepatic duct coming from the liver. We have the cystic duct coming from the gallbladder. They mix together, form the common bile duct, which moves downwards, where it's joined by the pancreatic duct. Ultimately, this purple part, that is everything combined, which is the hepatopancreatic ampulla of hepatopancreatic ampulla, or the ampulla of Vater. Right? And it is controlled, the emptying of liquids from this ampulla of Vater is regulated by the sphincter of Oddi. So all of these things hopefully are clear to you. If that is the case, we can start with some questions on this topic. Oops. Yeah. So in a liver lobule, blood from the hepatic artery and hepatic portal vein ultimately drain into which of these options? So I hope all of you are awake. You can try answering. Anyone who doesn't know, you can send the answer that you think it is into the chat box, okay? And then we can discuss. Anyone want to try? Okay, good. You're awake. I'm glad to see that. Um, yeah, you're good. You're awake. You're answering correctly also. This is very good. So it is indeed option B. You're right. This is the central vein. So inside each liver lobule, so blood is entering from like these branches of the hepatic artery, right? Like I mentioned, that's 25%. So you have oxygenated blood coming in from the hepatic artery, and you have a lot of nutrient rich blood coming from the hepatic portal vein. So normally they say that arteries will bring oxygenated blood to the organs and they will take deoxygenated blood away from them. But in this case, so typically arteries enter and veins leave, right? But in this case, the hepatic portal vein system is the opposite, right? So you have a vein that is connecting from the intestine to the liver, and that's why it's called a vein, because it's leaving one organ but it's entering into another organ. It's not going back to the heart. So anyhow, you have hepatic artery and hepatic portal vein both coming in, like branches of these major blood vessels coming into the liver lobule, and these mix into these little capillaries that are called sinusoids, and they flow towards the central vein, right? And that vein then ultimately drains into the vena cava and then it goes back to the heart. So it's like one major vein that is exiting. That's what the central vein is. So it's a central convergence essentially, and it's very important for the organization of the lobules in the liver. So yeah, that's the correct answer. Good job to you guys for getting it right. We can move to the next one. Okay, so which of the following correctly describes the direction of bile flow in relation to blood flow within a liver lobule? Yeah, great. So does anyone else want to give it a try? Okay, yeah. So good. The correct answer is absolutely C option, which is essentially that they flow in opposite directions. So one very important feature of the liver is that they have this counter current flow where the blood is moving from the periphery towards the central vein, and the bile, which is produced by the hepatocytes, that flows in the opposite direction towards bile ducts, which are at the lobule edges. And this is what allows sort of maximum efficiency when it comes to secretion and processing and stuff. So there's a counter current flow of bile versus blood in the liver. So great job. Good. All right. So about Kupffer cells present in the liver sinusoids, what is their main function? Anyone want to try? Anyone remember anything about Kupffer cells? They're very cool. Anyone else want to try? Yeah. So these Kupffer cells are, so the right answer here is option C, firstly, which are the phagocytic immune cells. So they are like basically a type of macrophage that are resident in the liver. So they're called resident macrophages of the liver. And they're quite specialized because they are, particularly for that organ. And they're very, very important for um, innate immunity maintenance inside the liver. They're found in the sinusoids only, these little capillary-like channels that we were talking about. And they sit over there, and then they're quite strategically positioned so that they can monitor the blood that's coming from the gut, right? Via the portal vein, there is this nutrient-rich blood coming, but that nutrient-rich blood could also have some bugs, right? Could also have some unwanted stuff. Um, so these cells are positioned very smartly such that the cells can interact with that blood, and there are detection mechanisms that could allow it to then activate some kind of response and itself phagocytose and kill any maybe bacteria that might be entering. So they have this like cleanup role where essentially they engulf and they destroy. So phagocytosis is what, right? It's a process by which a cell engulfs something else, right? Another cell, a piece of a cell, right? And it takes it within itself. So these Kupffer cells are a type of phagocytes, and so they're very important for picking up these bacteria and digesting them, or old and damaged RBCs, red blood cells, also, or any kind of cellular debris. Very important. It's a good cleanup function that the Kupffer cells have. And of course, they are the first line of defense when it comes to immune filtration for the portal vein blood. Yeah. So cool. Right. So that's about Kupffer cells. Hope that was useful for you. We can go to another question. So the contraction of the gallbladder so that it can release bile into the duodenum is primarily stimulated by which of these? So we've actually covered this before. So anyone want to try? Anyone wants to try? Yeah, I'm seeing some answers. Does anyone else want to try? I think I can move on. So the correct answer here, yes, indeed, it is option C, which is cholecystokinin or CCK. So CCK, which used to before it used to be called CCK-PZ, which is cholecystokinin pancreozymin, but it turns out that it it's just it's just CCK now. So this hormone is released, sorry, enzyme is released when fatty acids and amino acids, like a bunch of them, enter the duodenum, and it stimulates the gallbladder contraction. And that is what, so if it contracts, it's like a little bag, it's like a little balloon. So you contract it, and that causes the bile to be pushed out into the bile duct, and then that eventually carries into the intestine so that it's um, allows for proper fat digestion. So it's very important that bile needs to be delivered precisely when fat digestion is required. So the CCK is what helps the gallbladder to contract. Great. Okay, another question. So what is one of the major functions of bile salts in digestion? Anybody know? Anyone? Right. So the correct answer here is B option, right? Because what we talked about earlier was how important liver, and because of liver, the production of bile is possible, how important bile is for breaking down fats. So how, right? How do you digest fats? So one of the first steps is to emulsify them. So bile salts basically act like detergents. So they break large fat globules into tinier droplets, which are called micelles. And then that increases the surface area essentially that is available for lipase to then come and perform its action. So if there was no emulsification, suppose there's less bile or no bile, then fat digestion would be very, very, very inefficient because the enzymes cannot access such large masses of lipids properly. So that's why bile is very important, because it acts like a detergent and breaks down lipids into smaller particles called micelles. So the correct answer here is option B. Okay. So I hope that is clear. I hope the function of um, bile salts are clear now to all of you. All right. I think one last question before we move to the next topic. So what is really enterohepatic circulation? What does it refer to? Anyone want to try? What is enterohepatic circulation? Anyone? Okay. So, enterohepatic circulation, you might think it's relating directly to blood, but actually, it is with reference to these bile salts, right? So, it's the recycling of bile salts between the intestine and then to the liver. So, it's option B here. It's a very important loop, this enterohepatic circulation, that allows the body to reuse bile salts many times during digestion. So sort of how does it work? So first, in your liver you have hepatocytes producing bile acids from cholesterol, and then these are conjugated to form bile salts. Okay, so that makes them, if you're synthesizing from something from cholesterol, it's going to be very hydrophobic. So then you also add in some conjugations that make them salts, which are more water soluble. And then this bile is stored and concentrated in the gallbladder. And then if you eat a meal, particularly if it's a fatty meal, that causes the release of CCK, right? Cholecystokinin, which then stimulates the gallbladder contraction, which then releases bile into the duodenum, the small intestine. And then bile salts, as we talked about, they emulsify the fats, make them smaller, increase surface area, and so lipases are able to act on them and eventually break them down such that lipids and fat soluble vitamins like vitamin A, D, and K can all be absorbed. Now, there is a key step here, which is reabsorption. So, 95% of these bile salts are then actually reabsorbed in the last part of the small intestine, the terminal ileum. So, there's this active transport mechanism that allows for these bile salts, like a majority of the bile salts, literally, to be reabsorbed at the level of the ileum. And then via the portal vein, all of this goes back to the liver. And then it goes into the hepatocytes and then it's reused all over again. So it's like a reduce, reuse, recycle, this enterohepatic circulation. And for every meal, this can happen like 4 to 10 times apparently. That's what I read online. Yeah, cool. So I hope that's clear to you all and interesting for you. Yeah. Cool. So Right. Maybe we can just have a quick um, look at this, which is about the endocrine system. So, does anyone have any questions relating to anything in digestion or even the smooth muscle stuff that we talked about in the previous two classes, or anything relating to the liver or gallbladder or anything, stomach? Let me know before I move to this next topic. Okay, I think everyone is either fine or asleep. So, all right, let's start the next topic, which is the endocrine system, right? So, we all know about the endocrine system, right? It's the system that works via hormones, right? It produces and secretes hormones, and those hormones, even in very tiny quantities, regulate so many important functions in our body. Without proper control of hormones, regulation, feedback mechanisms, and so on, our bodies would go crazy. So, hormones are really important. And so these endocrine glands are the type of glands that secrete their products, which are hormones, directly into interstitial spaces, and they can be absorbed into the blood rather than going through any particular type of duct, which is why they're called endocrine glands. So the major glands, when it comes to the endocrine system, some are the pineal gland, the pituitary gland, this is all in the brain. Then you have the pancreas, the ovaries, testes, thyroid gland, parathyroid gland, the hypothalamus back in the brain, and adrenal glands on top of your kidneys. So the hypothalamus and pituitary gland are neuroendocrine glands. And studying this endocrine system, that makes you an endocrinologist. So sometimes when patients get sick and they realize that there's some, major hormonal issue, because maybe there's some problem with some particular gland that's causing either hyper production of a particular hormone, or it's leading to hypoproduction or lower production than required. So in this case, sometimes patients are referred to endocrinologists, people who are very highly trained specifically about how these kind of hormone affected diseases and disorders can be treated. Yeah. So some examples here of hormones: the thyroid, it secretes thyroxine. Pituitary gland secretes growth hormone as well as a host of other stuff. The pineal gland, which you may not have heard of too much, it secretes melatonin, and the testes secrete testosterone, the ovaries secrete estrogen and progesterone, right? So these are some of the very major and well known and well heard of hormones. I'm sure you've heard of most of these, right? And here they are. Here they're pictured, right? You can see the pineal gland, hypothalamus, pituitary gland, and then you have parathyroid, thyroid over here near the windpipe, the thymus over here, this little butterfly shaped gland where T cells go to mature. Then you have the adrenal glands sitting on top of the kidneys. You have your pancreas, that have the islets of Langerhans, very important. And then you have the testes in males and the ovaries in females. And these all work together. So, a little bit about some of the major components and contributors of the endocrine system and hormonal control. So, one is, of course, the hypothalamus, right? So, these are just some examples. I'm not talking about everything, but yeah, the hypothalamus is very important. Um, it sort of links the endocrine system and the nervous system. And you have different parts of the hypothalamus. Essentially, it makes two major hormones um, that then the pituitary gland stores and releases. So, oxytocin and vasopressin are very important. Um, and it also releases dopamine and somatostatin on its own. So, as you know, these are all highly essential hormones. You also have the pancreas that we just talked about, right? And that has, well, it has three hormones, but the major ones that we talk about our insulin and glucagon that work against each other. And deficiencies in either of them can be catastrophic. And then you have adipose tissue, which is your body fat. And you would think that it would not be secreting anything, but actually, as we talked about leptin a little earlier, and there's also other hormones like angiotensin and adiponectin and so on that are also released from adipose tissues. So this adipose tissue or fat tissue could be in many different places. Subcutaneous fat is right under your skin, between your internal organs, and inner cavity of the bones. So the yellow marrow has a lot of adipose tissue, right, towards the ends of the bones, right, where there's not much bone marrow, there's a lot of this fatty stuff. Yeah. Then there's the ovaries. They're the little oval-shaped glands in females on either side of the uterus, right? And that's very important for storing eggs and stuff. And these ovaries are like literally the center point of the maintenance of the menstrual cycle, maintenance of the correct hormones during pregnancy, and so on. And finally, you have the testes, which are the small round organs, and they are the ones that are particularly important for secreting testosterone, right? In mostly in males. Okay, so a few questions now about the endocrine system in general. So, what is another name for the pituitary gland? Do you guys know? Anyone want to try? Okay, so maybe I'll tell you guys this one. So, the pituitary gland is also called the hypophysis. So why is that? Because hypophysis means like an attachment below the brain. So it's actually anatomically located beneath the hypothalamus, and it's very, very important for endocrine regulation. So because of that, because it's below, it's called the hypophysis. Okay. All right. Next question. So the anterior pituitary develops embryologically from which type, which layer essentially? Do you know? This is sort of like a development question, like while development is taking place, where does the anterior pituitary actually come from? So, anyone want to give it a shot? This actually is what makes the sort of origins of the pituitary very interesting, embryologically speaking, like during development. So the right answer here is option C, which is an endodermal outpocketing of the alimentary canal. So what does it mean? Essentially, during development, you have, the endoderm, mesoderm, ectoderm, right? So most of the neural system is part of the ectoderm. But interestingly here, the anterior pituitary actually originates from and the endoderm. Okay, it's this outpocketing of the alimentary canal is called Rathke's pouch, and it is essentially an of endodermal origin. Whereas if you look at the posterior, so anterior means towards the front, posterior means towards the back. So the posterior pituitary actually arises from neural tissue, which is ectodermal. So this gives the pituitary a dual origin, right? Anterior is from one dermal layer, developmental layer, and the posterior pituitary is from another. And so that's why the pituitary could be called a composite gland because it has dual origin with respect to its different parts. Okay, cool. All right, next question. Right. So hormones like vasopressin and oxytocin are released from the posterior pituitary because of which of these reasons? Or more like how? So what do you guys think is the right answer for question 9? Anyone want to try? Okay, no problem. So the correct answer here is option C. So this vasopressin and oxytocin, they're synthesized in the hypothalamic neurons. And then after that, they're transported along those axons of those neurons, right? And then they're released into the blood at the level of the posterior pituitary. So this is like a neurosecretory mechanism. So that's how these hormones are released from the posterior pituitary. Yeah, so I hope that's clear. Cool. So let's move to another few questions and then we'll start a new topic. Right. So there is this portal system here also called the hypothalamo-hypophyseal portal system. So what is its primary function? So a portal system is essentially when you have two organs or two glands that are connected to each other and there's some, transport between them. So it's not just pure, oxygenated, deoxygenated, coming and going respectively. But there is some internal transport. That is when it's called a portal system, like you had in the liver when it was connected to the intestine. Here you have one between the hypothalamus and yeah. So anyone want to try? Yes, yes, good job. So it is indeed option B, which is that the hypothalamo-hypophyseal portal system. This is what carries hormones that need to be released from the hypothalamus to the anterior pituitary, which is the front part of the pituitary, right? And that allows a much more precise hormonal regulation without diluting it all in the system. So if this hormone was released from the hypothalamus and then it went all across the body and then went to the pituitary, or even if it went all over in the brain and then went into the pituitary, that would be not very optimal, right? Because it would be diluted. We'd have like, basically like clearance, loss during the circulation. So this direct transport between uh, the hypothalamus and the anterior pituitary of whatever hormones need to be released is very efficient and it prevents loss, it prevents systemic dilution. Cool. All right. Another question. So, which of these is not a hormone of the anterior pituitary? So, let me see if you guys know. So, the key here is anterior pituitary. This, oh, good. Okay. I'm getting some answers here. Yeah, great job, guys. So the correct answer is C option, which is oxytocin. So oxytocin is released from the posterior pituitary and not the anterior region. So in the anterior pituitary, you have secretion of hormones like TSH, thyroid stimulating hormone, ACTH, adrenocorticotropic hormone, growth hormone, luteinizing hormone, follicle stimulating hormone, right? ACTH, GH, LH, FSH, and prolactin. All of these come from the anterior pituitary, but oxytocin comes from the posterior. Okay, great. Good. Glad you know that. Let's move on to the next question. So, growth hormone, like I mentioned, exerts many of its effects, but it does so indirectly one way through which of these? Anybody, any idea? Yeah, good job. Absolutely. So the correct answer here is B option, which is IGF-1. So growth hormone can obviously act directly on tissues also, but it also can stimulate the liver to produce this IGF-1. So IGF-1 stands for insulin-like growth factor 1. Okay. And it's produced by the liver upon stimulation from growth hormone uh, from the pituitary, and this can mediate many growth effects like bone growth and tissue growth and so on. So growth hormone can actually act both directly and indirectly. Okay, I think a few more questions before we move to the next topic. So this is about somatostatin, right? So it's released from the hypothalamus and has what kind of effect on growth hormone secretion? Yeah, does anyone else want to try? Yeah, so perfect. You guys are absolutely right. It has an inhibitory effect, right? So it inhibits growth hormone release. So somatostatin will inhibit a hormone release, growth hormone release, whereas um, there is this GHRH, which is the growth hormone releasing hormone. I guess it's called GHRH, right? Yeah, growth hormone release hormone, I think, and that stimulates growth hormone release. And so there's like a dual control, right? So that allows quite a fine regulation of, when and how much of growth hormone should be secreted. Great. Okay. Um, Growth hormone is also called a diabetogenic hormone. So why is that? Could you guys see? Anyone? So think about like what growth hormone is trying to do in the body and what would be needed for those actions. So that will make it pretty clear. Yeah. So good. So the correct answer here is actually C option, because you have to remember what growth hormones purpose is, right? The purpose is for bone growth, tissue growth, and all of this, right? So what do you need when you're trying to grow stuff? Meaning you're trying to stimulate expansion of tissue, right? You're trying to stimulate cell division, right? You're trying to make new like tissues. So you need energy for that, right? So in that case, growth hormone actually increases blood glucose levels. It promotes glucose production, reduces glucose uptake, and it does opposite of insulin action. So that's why it's called a diabetogenic hormone. Okay. Final question. So in individuals that have normal growth hormone levels, but they have a deficiency of IGF-1, what is the expected outcome? What kind of condition would that person have? So think about how the growth hormone acts, right? There was a direct and an indirect way in which growth hormone acts in the body. So direct means it directly acts on the tissues, and indirect is it acts via a particular factor, right? This insulin-like growth factor 1, IGF-1. So actually, it turns out that the correct answer here, which is C option. So that means that only growth hormone, even if it's in the normal level, is not enough for proper growth. If there is a deficiency of IGF-1, if there is a deficiency of this insulin-like growth factor 1, which is produced by the liver upon stimulation with growth hormone, so it's an indirect way. And then this IGF-1 goes and helps with bone growth and tissue growth. If you don't have enough IGF-1, but you still have normal uh, growth hormone, it's not enough. IGF-1 is very essential for growth hormones effects, even indirectly so. So the person would not have normal growth, unfortunately. In such a case, they would likely have dwarfism. Yeah, great. So that was a couple of questions on this. Uh, yeah. So now we can talk about the thyroid gland. I think that was the last major topic covered in the theory that was uploaded, right? So you guys have all heard of the thyroid gland, I'm sure, endlessly. So the thyroid gland is found in the front of the neck, right, under the voice box, which is your larynx, and it has this butterfly-like shape, and it's yeah, the two lobes of this, right? So yeah, the two lobes of it here, they are on either side of the windpipe or your trachea. And this strip in the middle, that is a tissue strip known as the isthmus. So that's sort of the anatomy of it. And obviously, the thyroid gland produces extremely important hormones, T3 and T4 being particularly essential for basal metabolic rate. So you have T3 and T4. So T3 is triiodothyronine and T4 is tetraiodothyronine, and we also call it thyroxine. So these these two hormones, particularly, are very, very important. Um, okay. And then there's another hormone called calcitonin, though that is not considered a proper thyroid hormone. Calcitonin is made by C cells, another type of cell. And this is involved in calcium and bone metabolism. So basically, metabolism of different types, these are all um, highly regulated with the help of the thyroid gland. And you would notice that there's this iodo, right, in these names of T3 and T4. That's because iodine actually is a very, very important substance in this case, and it's needed to make these thyroid hormones T3 and T4. But actually, we don't produce any iodine, obviously, in our body, so we need to get it from food, which is why, salt was iodized and all of that, right? To make sure we're getting adequate iodine, even if it's via salt, into our body, because it's very important for the health of our thyroid and the correct amount of production of these thyroid hormones. Um, yeah, so that's why this iodized salt con concept was even thought up. Yeah. So, yeah, I had, I think, a few questions which we can just glance through now because I don't want to, drag on too late. Okay, so we were just talking about this, but where is the thyroid gland anatomically located? Could you guys tell me real quick? Yeah, good. So I've got a correct answer here, which is B option, which is on the trachea near the larynx, right? It was below the larynx. So it's located anterior to the trachea. So suppose somebody is, lying down on a flat surface, the part facing up, right? Up meaning like their, like in the, like where their nose is, right? That is the direction. So that's anterior. Um, so this is located anterior to the trachea, basically in front of it, and then just below the larynx. And yeah, it has this butterfly-like shape. It's connected by an isthmus, the two lobes of it, right? Sort of wrapped around the trachea, kind of like that. All right, so that was simple enough. There's another question. So the thyroid gland is unique among the endocrine glands. Why? Do you guys know? Anybody want to try? So this is an interesting one, right? Because it turns out that most endocrine glands, the cells actually store the hormone within them. But here, the correct answer being option C, actually the thyroid gland is quite unique because it stores the hormones extracellularly in this colloid-like form. So in the follicular lumen of the thyroid gland, um, the hormones are stored like outside of the cells in the basically like the ECM essentially, in this sort of colloidal form, and that's why it's quite unique. It's different from the other glands in that aspect. Okay. So another question. So which cells of the thyroid, I didn't discuss this, but we'll discuss it now, are responsible for secreting T3 and T4? Anyone want to try? What type of cell is responsible for secreting T3 and T4? Which type of cell here in the list? So there are really sort of two major types of cells in the thyroid, right? You have follicular cells and parafollicular cells. So in this case, which is this last question here, the cell type responsible is the follicular cells. Okay, so the follicular cells, which are essentially there are these little follicles inside of the thyroid, and these follicular cells line those follicles, right, on the inner side. And these follicular cells then synthesize T3 and T4 hormones, and they directly interact with this follicular lumen, which has this colloid, and that's where the hormone precursors are stored. So the answer here is the follicular cells. Okay, they are responsible for secreting T3 and T4. The follicular cells, I think, are also called principal cells. They're like a wall um, in the thyroid follicles. Oh, by the way, the colloid inside of these follicles, I forgot to mention, is it's called thyroglobulin, in which this these hormone precursors are stored, and then those go through these follicular cells, and they come out as final T3 and T4 or triiodothyronine and thyroxine. Okay, good. So next question. So calcitonin, which was the third type of hormone that we talked about, right? What is that secreted by? So this makes it pretty clear considering there are only two major types of cells. Anyone want to try? Yeah, awesome. So it is indeed C option, which is the parafollicular cells. And they secrete, so the parafollicular cells are also called C cells because they secrete calcitonin. C for calcitonin, so C cells. And this peptide is very important for lowering blood calcium levels. So by reducing blood calcium levels, so how does that work? You can actually, if it inhibits osteoclast activity, it reduces bone resorption, so it decreases blood calcium levels in that way. Yeah. All right, let's do another question. All right, so T3 and T4 are derived from which amino acid? I wonder, can you guys tell me? Yeah. Yeah, great. So the correct answer is B option, which is tyrosine. So basically, they're synthesized from both of these guys, T3 and T4. They're synthesized from tyrosine residues, after which, like that base, whatever structure is then iodinated, as we, learned about how important iodine is. So it's iodinated and coupled, and that's what forms the final thyroid hormone. So it's not so simple, there's actually a lot of steps in it. It's quite tightly regulated by TSH, which is your thyroid stimulating hormone. So a few steps involved here is that one, obviously, the follicular cells where this stuff is all happening, they need to have iodine in some way, right? So there's actually this interesting symporter that's found in these follicular cells that takes up iodide from the blood, right? I minus. So it's like it traps the iodine in the form of iodide. And this is very important. It depends on energy. And then this iodide is moved into that thyroglobulin colloid in the follicular lumen, and then this iodide is converted into this active iodine form, and there's some enzymes and all involved in that. And then this iodine binds to the tyrosine residues that's found in thyroglobulin, and it forms some complicated structures. And this also needs like enzymes and some level of energy and stuff. And then there's a whole bunch of coupling reactions that finally give these iodotyrosines and give this T3 and T4 final product. But this is still attached to this colloid, to thyroglobulin that's in the colloid, right? It's still attached to that. And it's stored in that form extracellularly, right? Until there is some TSH stimulation, some thyroid stimulating hormone is released and that stimulates. And then this colloid is endocytosed and thyroglobulin is cleaved off, and T3 and T4 are free to be released into the bloodstream. So until then, it's nicely held, sequestered in the colloid in its iodinated form, in its full ready form. It just needs to be cleaved and sort of released. So TSH helps with that process. Otherwise, all of it will be retained. Yeah, cool. So, all right, let's do a few more questions and then we'll be done for today, I think. So, which of the following correctly describes the relative abundance and activity of thyroid hormones? So, which one is more abundant? Which one is more active? Do you guys know? And like why, maybe? So this is interesting, right? Yeah. So, good job answering. So, you are absolutely right. It is option B, which is that the T4. So, it's very interesting, right? So, T4 is more abundant than T3, but it's less active than T3. So it turns out that 93 or approximately, 93% of circulating hormone is actually T4. But while T3, which is only like 7%, right? It's less abundant, but much more biologically active. And one of the reasons is that it's often formed because it just converts from T4. So you can get T3 from T4. So you just have a huge bunch of T4 that's less active in the blood, and then some of it is actually converted to T3 as in when required, I suppose, and that is less but more biologically active. So maybe you can, guys, if you're interested, could read about why that is the case. Why is it that something that is less available is more potent? But that would make sense, right? If you want to, if you just think about, say, any mechanism where you have one thing that is like highly active, if you have too much of it, that could lead to problems, right? That could lead to dysregulation, that could lead to overactivation. So you would want to have a controlled amount of something that's very potent. Whereas if you have something that is less potent or less active and you have more of it, then there's less chance of off-target hits. There's less chance of dysregulation. So maybe that's why, right? Okay, last three questions. So, yeah, so I talked when I was talking about the synthesis of T3 and T4, there was this iodine trapping mechanism in thyroid cells that I mentioned, right? Briefly. So, what does this primarily depend on? Yeah. So, yes, the correct answer here is option B, where there's a sodium gradient via this NAK pump. So, iodine uptake um, depends on this energy driven sodium iodide importer, right? And this is powered indirectly by a gradient of sodium that's maintained by a sodium potassium ATPase, right? So that we had read about, right? When we looked at different types of transport, that there is this type of transport where you have a gradient maintained by another set of ions that allows one different symporter or antiporter to function. So an electronic or electrochemical gradient that's maintained across um, one type of ion can actually help fuel the transport of a different ion. So yeah, in this case, it's the sodium iodide symporter that is powered indirectly by the sodium gradient maintained by the sodium potassium ATPase. So yeah, you're absolutely right. The answer here is B option. Okay. So then there's this thyroid peroxidase, TPO. Okay. And this is essential for which um, of these steps? I didn't mention it earlier, but we'll discuss it right now. Yeah, so you guys are absolutely right. It is option B, which is um, oxidation and organification of uh, iodine. So essentially, it catalyzes. This is a TPO is an enzyme, right? So it catalyzes oxidation of the iodide, and then that allows its incorporation into something organic, which is why it's like organification into the tyrosine residues, and it's a very, very important step in the hormone synthesis process of in the thyroid. Okay, and the last question for today, I'm glad we managed to finish everything. Um, the thyroid hormone increases basal metabolic rate mainly by which mechanism? Yeah, so you guys are absolutely right. It is option C, which is that there's an increasing metabolism, right? So the thyroid hormones increase the basal metabolic rate or the BMR by increasing or enhancing oxygen consumption and like mitochondrial activity, enzyme synthesis. It affects nearly like all the tissues in the body. They're really, really important. So it's what even is BMR, right? So basal metabolic rate is like the minimum amount of energy, the basal amount of energy required for all your essential function like breathing, circulation, ion transport, cell metabolism, whatever, right? So if one were to measure it, it is the oxygen consumption at rest. Okay. So how do thyroid hormones do this? So for instance, T3 in its active form, it will enter cells, it will bind certain nuclear receptors. That will then cause gene transcription of various metabolic enzymes, and then you have more of the enzymes, more activity, higher BMR, right? There's also a heat production. So there's this calorigenic effect where this the thyroid hormones can increase the activity of sodium potassium ATPase, increase ATP breakdown, and that releases heat, which is why people, the patients who have hyperthyroidism, too much thyroid hormone, they feel this heat intolerance kind of thing. So essentially, they increase oxygen consumption, these enzymes, right? Sorry, the hormones, and they increase mitochondrial activity, more heat, right? More metabolic activity, more nutrient turnover, so more glucose absorption, more lipolysis, more protein turnover, whatever, right? So that's what that's what these hormones do, but obviously in a regulated manner. So in the case of hypothyroidism, you would have the BMR going low, and in hyperthyroidism, the BMR would go higher than normal. So in hypothyroidism cases, for instance, people uh, undergo weight gain, cold intolerance, right? They feel cold. This heart rate is slower. They feel fatigue because they just don't have enough energy. And hyperthyroidism, essentially, that can cause a lot of weight loss. Despite your appetite increasing, weight is just being lost because ATP is just being, it's just being broken down and like consumed like anything. And then there's heat intolerance, sweating, tachycardia. So all of this is basically like one is making you like, burn like crazy, and the other is making you sort of slow and cold. Right? So that's how important BMR is. Great. So I think that's all the questions I have for today. I hope this session was useful to you guys. Um, you can let me know if you have any questions, any further questions. Um, yeah, and we can then meet in the next class and discuss more topics then. Yeah, so yeah, no problem. Yeah, hope it was useful for you, right? We'll see you next class then. Okay, all right. Bye, everyone.